So on behalf of the graduate school, I'd like to welcome everybody to the first in the spring semester series of Provost Graduate Student Lectures. We have a really very interesting schedule this fall of this spring of, I think, 6 lectures that celebrate the not only the breadth but the excellence of the academic achievements of graduate students through all divisions of the university, from STEM to the fine arts. To kick the series off, today's speaker is Georghiou from the Graduate Program in Molecular Pharmacology. And I'm going to let his advisor, Dr. Marcus Sellinger, tell you a little bit about George before he talks.
Thank you, Joel, for introducing George, and thank you very much for giving George the opportunity here to present. George did his undergraduate degree in pharmacology here at Stony Brook, and then he hung around to do his PhD in pharmacology, and that's where it's about to end because he's going to leave Stony Brook after his PhD very soon. But it's a great opportunity for George to tell us a bit about what he's been doing in my lab. What he's not going to talk about is his first research project, and that's basically finding the ways of unpacking all the stuff and setting up my lab and finding all places then to store all his things in the lab. So this is not part of today's talk, but George is going to talk about some very productive and very exciting work that he has been doing in collaboration with David Liu at Harvard University initially. And then secondly, he's going to talk about another project that is entirely based on work that George initiated in the lab. So, and with that, I'm looking forward to hearing what George has to tell us.
Hi, everyone, and thank you for coming, and thank you to the organizers for giving me the opportunity to present here. So, the title of my talk today is Hitting the Bullseye in Cancer Therapy. What exactly we're going to be discussing today is some of the new approaches and therapeutics that we've been using to battle cancer in the clinic. As we all know, cancer is a pretty severe disease, one with a high rate of death, and has only been on the rise. The number of new cases of cancer has only been on the rise over the last, well, as our quality of life improves, we live longer, and the risk of developing cancer later on actually raises as well, because age is the #1 risk factor for cancer. And within the there was just a report released by the World Health Organization that stated within the next 30 years, the number of cases of cancer will rise by 17%. And this is the new cases of cancer, meaning we go from 17 million up to 40 million. Wait, is that right? Yes. Oh, sorry. Yes. Alone in the US, currently within at least 2012, we had over 1.5 million cases of cancer, new cancers reported. Within the next 30 years, we expect the number of cases in cancer to rise up to almost 2.5 million per year. This is saying pretty much that in 2012, the entire population of Suffolk County developed cancer. And then within the next 30 years, we're going to tack on the population of San Francisco to this number. So it is actually a bit of a problem that we have set before us, and developing new therapies is actually critical for this. Almost 2 decades ago now, a drug called Gleevec came out on the market. This was very different than a lot of the chemotherapeutics used in the clinic because it was what we call a targeted therapy. In fact, this drug was so revolutionary that it reduced the number of deaths related to the leukemia it's used to treat by 80%, and is actually still used as a frontline drug in the clinic. And we've been trying to repeat the success of imatinib with other generations of these particular types of drugs that I'm going to be talking about. We refer to them as kinase inhibitors. Dasatinib and nilotinib were second-line drugs that are now being currently used for a different number of diseases, for a different number of cancers, in particular different leukemias, gastrointestinal tumors, and actually more recently, we're starting to take a look at these drugs as potential breast cancer therapeutics. So going off how imatinib really was quite successful in reducing the number of cases of leukemia, reducing the number of deaths related to leukemia, drug companies have spent the last 20 years developing new inhibitors to come up with a new drug, a better drug than imatinib for different cancers, to say, can we pretty much eradicate cancer? So the purpose of my thesis initially, in collaboration with David Liu at Harvard University, was to see if we can build on the success, making more specific inhibitors. And the second half of my project was looking at kinases, which I'm going to talk about in a bit of what these molecules are, and how can we discover kinases that seem to promiscuously bind a lot of these drugs, and how that may affect the effectiveness of chemotherapy with these drugs. So what are kinases? Kinases are signaling molecules that all our cells have. And they're like molecular switches, pretty much. They turn things on and off. How do they do this? Well, kinases utilize a substrate known as ATP. And our cells have a lot of this. It's not only as part of the signaling molecules, also the energy source of the cell. And what kinases do is they take a protein substrate, another protein that happens to be nearby, by proximity, or just something a kinase will target that needs to be phosphorylated. That's the event that kinases seem to regulate. And what they do is they take ATP and they convert it to ADP. And they add on a phosphate group to a protein substrate. And what happens there is that that either turns off or on that protein substrate, and what we get typically is a cellular response. So the only other thing about kinases is that there are actually about 518 of them coded in the human genome. There's a lot of them, and they all look very similar, at least in terms of the kinase domain that I'm going to be talking about today. And on top of that, they're incredibly tightly regulated because the cell is a coordinated system. You can't have things turning on and off all at the same time because it's going to lead to a lot of different problems, potentially different diseases. And we give this analogy as, flying a space shuttle. If you want to land a space shuttle in space, from space onto the ground, you can't flip on everything at once and expect it to end well. If you turn on the engines without actually controlling which direction you're going in, you're going to crash. And in cells, if you turn on everything at once, you're most likely going to develop a disease. And that's why it's not very surprising to find that kinases are dysregulated not only in cancer, but also in diseases relating to chronic inflammation, like rheumatoid arthritis, hypertension, and Gordon's and Gitterman syndrome, hypotension and Gitterman syndrome, and neurological disorders like Parkinson's and Alzheimer's disease. So this really asks the question, how can we inhibit dysregulated kinases? So imatinib is a small molecule inhibitor. It's what we refer to as a small molecule inhibitor. And as I said before, drug companies are trying to come up with more successful drugs like imatinib to treat different kinases, to target different kinases out of that whole kinome. That's what we refer to this large family over here. And there's two approaches to doing this. There's the shotgun approach, where you develop something that can just target multiple kinases and hope for the best that it works. Sometimes it's okay. Most of the time it's not. We really want to target one, which is a targeted therapy, where we aim at one kinase in particular that's responsible for one particular disease, let's say. And that's what we're trying to do here. And this is really quite challenging because they all look alike. So these drugs have to be incredibly specific. The other thing that a lot of these small molecule inhibitors have to overcome is affinity. All kinases use ATP. And the cell has tons of it. This is actually not a two-scale representation about how many molecules of ATP your cells have, but it's just to give you the idea that a single drug molecule has to compete with a ton of ATP for the same kinase. So these drugs have to not only be specific, but they have to bind more tightly than ATP. So we've taken a number, at least in the field, there have been a number of different approaches to develop kinase inhibitors. I've already mentioned to you imatinib and dasatinib, really briefly before, that are ATP competitive inhibitors. This is the active site of the kinase, and they occupy the site where ATP would bind. Another method that's been used is peptide competitive inhibitors, pioneered by Andrew Levitsky's group in the early 90s. They were referred to as the tyrosines. They were incredibly selective because each kinase has a select number of proteins that they can target. So they figured they can generate more specific inhibitors this way. The only problem was that these inhibitors didn't really work so well in cells. In fact, they were not as potent as the ATP competitive inhibitors. And finally, there's this group of drugs known as the allosteric inhibitors that target other parts of the kinase that are not related to where ATP and proteins bind. And we know very little of these sites in kinases. In fact, a lot of work has been put into finding them. And I think currently in the clinic, there's only one allosteric inhibitor currently used. So a lot of the focus has been working on the ATP competitive versions. So just to continue on with a little bit of the background of kinases, this is what a kinase looks like, more or less. This is where all the catalytic activity takes place. It's a bilobal structure, having an N lobe and a C lobe. And in between the two is the active site that I mentioned before. Where my laser is shaking, you see that's where ATP binds. And over here by the purple element over here is where protein would bind. I highlighted here 3 regulatory elements in kinases that let us know whether they're on and off. And I'll go into a little bit more about these a little later, but I'm not going to talk about it right now. Now, kinases, as I said, they can turn things on and off. And we know kinases themselves have at least one on position and 2 off positions. And the differences are noted in the orange and blue elements highlighted here. We have drugs currently like the dasatinib that I mentioned before that can target the on position. And as I also mentioned before, imatinib, which can target one of the off positions. The only thing is we don't really have any drugs that bind to this off position for kinases. And it's actually something I think has not really been pursued in a lot of different in terms of developing drugs for kinase inhibitors. And it's something that I will touch on now. So going on to the first part of my project, which was developing these specific inhibitors for kinases like Gleevec, we used the model system of the Src family kinases. Now, drugs like the dasatinib bind to Src, and they also bind to this kinase over here, Abl kinase. Abl kinase is the actual target for imatinib or Gleevec. Now, the common convention is any inhibitor that inhibits Src will also inhibit Abl, but not vice versa. So we don't have any Src inhibitors. But the other thing about this is that there are no inhibitors that can actually tell Src apart from its family members. These 2 proteins right here, HCK and LCK, both kinases that can be found in a number of different lymphomas and leukemias to be mutated. And we don't have a drug that can tell the difference between either of these. So the Src family kinases, there's no way to differentiate them when you're inhibiting with a kinase inhibitor like the dasatinib. So we decided to use David Liu's compounds that I'm going to talk about in a second to kind of find if we can develop a specific inhibitor for Src, but can also distinguish it from the really closely related HCK kinase and the LCK kinase, and then of course not inhibit Abl at all. And through a very clever way of synthesis, David developed these cyclic peptide inhibitors. We refer to them as the MC compounds. And they have different chemical groups in the A, B, and C position. Different chemical groups actually affected how well these drugs bound to kinases, and we'll show you some of the data that we have for that at the moment. This is MC4B. It contains, as you see, different chemical groups in the A, B, and C position, highlighted in red, blue, and green. And what I did here is I looked at how well they can inhibit the kinases that we were looking at, Src, HCK, LCK, and Abl. The IC50 is just a measurement of how potently these drugs can block kinase activity. And the lower the number is, the better. So in the case of Src, we see that it binds with a really, really low IC50, so this is great. And we see that for HCK and LCK, the compounds have reduced potency. And then for Abl kinase, they don't work at all. So we determined, based on just potency alone, that these compounds were specific to Src, and were able to differentiate Src from the Src family kinases, as well as Abl kinase. Now, it's great that we have a selective drug, but the most important question we're usually asked is how do they work? So what we did is we actually took a look at how these where potentially these drugs may bind. And what we realized is that when we do binding studies to see how tightly these compounds can bind to Src, this is MC9. What we did here is we found out how tightly it binds to Src, and again, the lower the number, the better. This is a KD, it's just an affinity constant that we determine. And when we added substrate peptide and an ATP analog, we noticed that the binding of the drug was reduced. So to us, that demonstrated something actually really cool, because we now have an inhibitor that's both ATP competitive and peptide competitive, the two substrates for kinases. So this is great. This is actually a really unique approach because no one had really accomplished this so far. Now, our group's a structural biology group. We specialize in a technique called protein X-ray crystallography, and the most beneficial thing of this is that you can actually get a visual representation of what these drugs look like bound to a kinase. So how do we go from a crystal of a protein all the way to our three-dimensional structure? Well, as I said, you can crystallize a protein as implicated by the name. Shooting it with a high-powered X-ray allows us to get a diffraction pattern. This diffraction pattern can be through a series of mathematical formulas and really fancy software that I will not bore you with, can lead us to the development of a 3D structure. And I'll go ahead and show you some of the stuff, two of the structures that I solved for SARC bound to these MC compounds. Let's see if I can get this to work. So this is our structure of SARC bound to MC4B. This is, again, the bilobal structure that I showed you before, and these three regulatory elements that I had mentioned earlier. Now, these will determine whether the kinase is in the on or off position. And it just so happens it binds to that one particular off conformation, that one off position that we didn't have drugs for. So that was really a great benefit because not only do we have a bisubstrate competitive inhibitor that's specific to SARC, but it targets an off conformation that we haven't seen before. There's our drug in the active site of SARC. And if we actually take a look at how it binds, it explains the competitive nature of it. We see that one group is occupying where ATP is binding. The blue group is occupying a pocket that's only accessible in that one particular off conformation. And the green group is binding where the protein would actually bind itself. So we see that it explains why it's ATP competitive, because it's occupied in that position, and peptide competitive. This is SARC bound to MC1, another structure that I solved early on in my PhD. And we noticed that the larger the chemical group in that third position, if we were to model in a protein binding to a kinase, it would directly clash with it. So the larger the chemical group here, the more peptide competitive it is. Now, the more important question we asked ourselves, though, was, okay, we can explain the competitive nature of these compounds, but how do we explain the selectivity for SARC over HCK? How do they figure out which kinase, how are they specific to SARC in particular? And the structure, when we compared SARC to HCK, showed us 3 amino acids that were unique to SARC: glutamine 275, leucine 297, and cysteine 277. So what I went ahead and did was mutate each of these residues to the corresponding residue in HCK. So here's our data for MC4B. Again, this is just regular SARC. We see that it really is potently inhibited, HCK not so much. And when we change glutamine 275 to a glycine, which just removes this large blue group over here sticking out, we see that the potency of the compound is drastically reduced. So as the IC50 has now increased. Mutating cysteine 277 over here by this green group, that peptide competitive group, to a glutamine, which is a larger chemical group in this position. Although the data here shows that there wasn't a really drastic change in the potency of the drugs for this mutant, with the data that we had for other compounds, like that structure of MC1 that I showed you, with the larger group over here, this mutation had a greater effect. Because MC4B has a smaller chemical group over here, the effect wasn't as noticeable. Mutating leucine 297 to methionine. What I did essentially here was take this leucine, this little pocket that this blue group is binding in, and added a huge amino acid into it, which reduces the size of it. And that also affected the potency of the compounds by about 2-fold. Finally, we realized something about glutamine 275, which was glutamine 275 doesn't actually face anywhere near the drug. Actually, it faces the opposite direction. It faces up into the kinase, not into the active site where the drug is binding. And very curiously, it seems that SARC was a bit unique in that it contains this extra residue, this extra amino acid facing away from the active site that actually is part of an interaction in a conserved salt bridge between a lysine and a glutamate that's found in a lot of different kinases. So when we went ahead and decided, okay, is it an effect of just an additional interaction? Well, we can use an inhibitor resistance mutation that we've seen commonly for Abl kinase with imatinib, which is mutating glutamate 280, which would break the interaction over here to a valine, which is a much smaller group that can't interact with these two. And we also saw a reduction in the potency. So for this, it was really great because we managed to sum up, for the most part, that we have inhibitors that were selective for SARC, these MC compounds, that were bi-substrate competitive, and now we have a reason for why they're selective. We have 3 residues, well, glutamine 275, cysteine 277, and leucine 297, that'll provide a unique environment for these, that provide an environment unique to SARC for these drugs to bind to. So just to summarize, they're SARC specific, bi-substrate competitive, and now we target this one particular off conformation that I actually forgot to change the name over here, my apologies, that we had not targeted previously. Now, for the second half of my thesis work, we are looking at kinases that promiscuously bind kinase inhibitors. This is a bit of a problem because, as I said before, developing specific inhibitors can be quite challenging because the entire human kinome looks very similar to one another, at least the region that we're targeting for drugs. And in therapies, if you have a kinase inhibitor that binds to a lot of these, normal cells that are not mutated and cancerous can actually be affected, and it can lead to a lower quality of life for the patient undergoing treatment. So rather than make the patient miserable, we want to make more specific inhibitors, but in order to do that, we also have to understand the opposite perspective of the argument. We can develop inhibitors that are really specific, but we also need to understand why there are kinases out there that may bind these non-selectively. Lucky for me, there was a study done by Ambit Biosciences, a company out in California, that screened 372 human kinases against 38 kinase inhibitors, most of which were used in the clinic, others that were in clinical trials, and then finally others that were just used for research. And sifting through the data, we were able to kind of figure out, well, which kinases are more promiscuous than others for binding these small molecule inhibitors. So we started with a really broad drug and said if we can start from really broad and work our way down to more specific drugs, ones that hit less kinases, we might be able to reveal something a bit interesting and find a particular kinase that is more promiscuous than others. So the drug staurosporin, it's not a clinically used inhibitor. It's more a research purpose inhibitor. Actually inhibits a lot of the human kinome. And we found 176 kinases in the data that bound staurosporin with a particular KD, that affinity that I was talking about before. So I said, okay, that's great. Now from this 176, if we go to a more specific drug, not that specific, but something like dasatinib, which inhibits less kinases staurosporin, but also still you get a lot of non-selective inhibition. We found 24 unique to dasatinib and 22 in common with staurosporin and dasatinib. Going down a little bit more focused through the data, again, sifting through literature data, finding this. Like a drug like imatinib, which is incredibly selective, found out of all these, 7 kinases seem to be in common with that. And then, of course, I got this backwards and forgot to change it once more, so sorry, Marcus. Went to another drug that's a little less specific than imatinib, but more specific than dasatinib, this drug sorafenib, and we found 6 kinases that seem to bind all 4 of these inhibitors rather tightly when we compare it to some of the other kinases. And those 6 were DDR1, DDR2, KIT, PDGFR-A and B, and CSF1R. Now, what was really not that surprising to me is that dasatinib and imatinib and sorafenib were all inhibitors pretty much designed to inhibit PDGFR-A and B and KIT kinase, because these are known cancer targets. These are ones that we knew in the clinic were mutated in a number of different cancers and they wanted to develop specific inhibitors against. Well, they did, and well, 3 of these were really not that well known until more recently. TSF1R, which is colony stimulating factor 1 receptor, was recently found to be implicated in a number of different cases of Alzheimer's, as well as I just recently found out last night, breast cancer. So we had these 2 kinases sticking out, DDR1 and DDR2. No one really knew much about these 2 kinases other than that they were expressed in cells and they dealt with cell adhesion. But no one knew what their role in disease was. So it was only until they actually started looking at a number of different cancers because they kept seeing these guys pop up in all the inhibitor screens for all these new inhibitors that they were developing, that they realized that DDR1 activity may be dysregulated in a number of different cancers as well. But what is DDR1? It's what we call a receptor tyrosine kinase. It's part of this group over here, known as the insulin receptor kinase family. And they bind to collagen, extracellular collagen. And what this means, what is implicated by this, and what I didn't mention before, is that normal cells, when they bind to collagen, it lets them know that you're attached to something. If a normal cell detaches from collagen, it's not very happy. It automatically assumes, oh my God, I'm dying, I need to undergo cell death. So cells can automatically end their own, can undergo a programmed cell death procedure if they find themselves detached. But in cancer cells, they find they no longer need this. And this is what actually allows, this is part of why we have metastasis in cancers, where cells will migrate from one area in the body to another and develop tumors in other regions. And DDR1, not going into too many details about this boring pathway, seems to be in charge of letting the cells know that it's okay to stay alive or divide or even migrate if it has to, like blood cells, for example, use this to migrate through bone marrow and whatnot. But still, it isn't a driving cause. It isn't really a driving factor behind cancer. It's just one of these that happens to be carried along for mutations, it seems. And none of these drugs were designed to bind to it. So really the question is, why is this one promiscuous? Why is DDR1 promiscuous? And what can we do to avoid hitting it when we develop these new kinase inhibitors? Well, when I expressed and purified the protein in the lab, I did a little bit of a biochemical characterization, looking at how well ATP binds to it, this common substrate that all kinases use. And what I found out was that the Km is actually rather high. A good Km, a kinase typically binds ATP with about 50, in the tens of micromolar for ATP, so it has a very low Km. And in the case of DDR1, we're almost, in some cases, about 10 times more than what it should be. And if it can't bind ATP very well, and a lot of these drugs are competing with ATP, it means that drugs don't have a lot of competition for binding to DDR1. So that may explain part of its promiscuity. Because it's not able to bind ATP well, it may bind these drugs better. Now, taking a similar approach like we did with SARC and some of the MC compound inhibitors, we tried to crystallize DDR1 in complex with some of these small molecule ATP competitive inhibitors to see that maybe if the structure could give us an answer of why it may be promiscuous. Lucky for me, I was able to crystallize with two different inhibitors. The Aurora kinase inhibitor VX680, which was supposed to be selected for the Aurora kinase as an able kinase, but also bound DDR1 quite tightly. And the kinase inhibitor that I mentioned before, dasatinib, which is currently used in the clinic. And these are what the crystals look like. On the previous slide, you saw those as well. They're not always pretty, but they give you these nice little diffraction patterns that we can translate into structures. So here's our structures of DDR1, nice little movie for you guys. So DDR1, like SARC, bilobal structure, same regulatory elements that I highlighted before for you that lets us know whether the kinase is on or off. Here's VX680 bound to DDR1, and we notice it's again in an off position, which is a bit odd because VX680 is supposed to bind to an on position. So we thought to ourselves, are there any unique interactions? And it turns out that VX680 makes the same exact interactions with DDR1 that we see in other structures of it bound to Abl and Aurora kinases. So that doesn't explain the promiscuity for it. It actually makes the same contacts. We're going to switch over to our structure of dasatinib in a second, which is going to be in green. And you'll see a little bit of a difference here. Again, we find dasatinib bound to DDR1 in this off position, which is a bit odd because dasatinib too was supposed to bind to an on position. Again, same question, does it make any unique interactions with DDR1 that may explain its promiscuity? And the answer is no. It makes the same exact interactions that we see in other structures of dasatinib bound to different kinases. I'm going to have to pause this here for just a quick second, just to point out two things. If we take our two structures, VX680 in gray, dasatinib in green, two things, at least to me they stand out, and hopefully I'll point them out well enough to you to see it as well. Two things seem to move, depending on the drug that we have bound. If we pair gray to green, if we look over here in particular, we notice that in the green structure, it's pushed up a little bit more. And over here, in this loop, in green, compared to gray, has been pushed down. Very oddly enough, it seems that all the structures that we have of DDR1 show up with all the drugs bound in the off position, which for some of them was expected, for others not, in particular the drugs that I looked at. But the more curious thing was that the bigger the drug in this active site, the more drastic this shift was. And to us, we translated that as potentially being the kinase being more flexible than others. And by having this additional flexibility, the drug may be promiscuous because it just allows all these different types of inhibitors, regardless of how they bind, to fit into its active site, simply because it could just open itself up a bit more to accommodate them. The structure also was a bit useful for revealing to us why it's so inactive, why it had such low activity. And what we notice is that there's internal interactions with this particular loop with the bottom C lobe of the kinase. Now, something I failed to mention earlier was DDR1 activity in cell is really slow. I mean, mind-numbingly slow. In the fact, if we compare it to other receptor tyrosine kinases, we're talking about a time scale when we look at a good receptor tyrosine kinase, as soon as it binds whatever it's supposed to bind, gets turned on within seconds. DDR1 takes 8 hours. 8 hours, which is very unusual. It's a very slow kinase, very poor, and that also relates to its poor affinity for ATP. So we thought, at least I thought in this case, that additional interactions within the kinase keep it in this off position. And that's why all the structures seem to be showing up in this off position. And having these additional contacts means switching to an on position is harder. You need more of an input signal to make it switch over. So just to summarize this end of this project, we co-crystallized DDR1 bound to dasatinib and VX680. We did some biochemical characterization where we showed that it has very poor affinity for ATP, which may explain part of its promiscuity because it just likes to bind ATP competitive inhibitors than it does its own substrate. And furthermore, the structures may actually lead to the actual cause of the promiscuity, which may be due to the kinase flexibility, because it simply can just open itself up more to accommodate different drugs. Now, with that, I'd like to thank my advisor, Marcus Seeliger, my lab, in particular, Jovan Fregoso, Michael Pekowski-Gross, and Lisa Malone, who really helped me out with the MC work, the Miller Lab for their help on the DDR1 project, as well as my thesis committee, Todd, and of course, our collaborators at Harvard University, and all of you for coming and listening. Thank you.